Hello, my name is Conor Anderson, and today I will be presenting to you guys on my internship for my Master's in Science in Biomedical and Health Informatics. So the research project that I was involved in was a collaboration between NYU Langone Health, Baylor College of Medicine, and the Center for Discovery, who's my employer. The project was an IRB in identifying biomarkers of gastrointestinal comorbidities in autism spectrum disorder. It was a longitudinal pilot study linking multiomics with behavioral and gastrointestinal comorbidities. The research question that we were looking to answer was an investigation into the microbiome gut-brain axis in autism spectrum disorder, looking to identify whether specific microbiota have an association with better outcomes in relation to behavior and GI comorbidities. It was a longitudinal study into the effects of dietary and behavior interventions implemented at the center. We collected biosamples, food logs, behavioral and GI validated screeners at baseline, which was before entry to our program, and at 3-month intervals for the next year. The samples included stool, urine, and blood, and these stool samples were sequenced using 16S rRNA amplicon sequencing. The food logs collected 2 weeks prior to each sampling period. The screeners that we used included the aberrant behavior checklist, the repetitive behavior score, the room 4, and GI severity score. The approach I took, first of all, was to create a set of Microsoft forms to collect the screener data, and then develop a pipeline in Python to process and score the results of each of those screeners, and utilizing repeated measures and mixed effect models in R, analyze the longitudinal changes in the relationship between the variables captured in those screeners and the food logs. Finally, then I built a pipeline in a framework called Data2, also in R, to process the rRNA amplicon sequenced data, produce the amplicon sequence variant tables. That then allowed me to analyze those ASVs using another framework called CHIME, which was in Python, to summarize the taxonomy of the samples and investigate the alpha and beta diversity, as well as the relative abundance of the microbiota in the sample set. Next, I'd like to share with you guys some of the results using the repeated measures, linear mixed effects models, and relative abundance. So we can see here the repeated measures. In this example, we were looking at the lethargy score within the sample set, and we did see a significant difference between baseline and 3 months, and baseline and 6 months after entry into the Center for Discovery's program. Then looking at the linear fixed effect model, we can see with the forest plot here that there was an association between total dairy and a higher repetitive behavior score, as well as a trend towards a lower repetitive behavior score with higher fruits, grains, protein, and food dietary diversity in general. This is also working with quite a small n, so as the group increases in this ongoing study, we would expect to see more significance. And finally, this is the relative abundance of microbiota in the samples, looking at the family level taxonomy. And we can see here that Lactospiraceae, Bacteroideaceae, and Ruminococcoaceae were the top 3 bacteria found in this sample set. Some of the limitations that I ran into was with the CHIME tool. It had all been written in Python 2.7, and which is really no longer supported widely. I tried a number of methods, including using virtual environments to try and run the tool in Python 2.7, but a number of the dependencies, including NumPy, which is fundamental to, you know, carrying out this kind of analysis. Just no longer supported Python 2.7 installs of any kind. So as a workaround, I set up a virtual machine in Oracle VirtualBox, running a very old version of Ubuntu and Python 2.7, and was then able to use that to summarize the taxonomy and analyze the diversity and abundance within the sample. The next steps, we're targeting publication of this research, along with analysis from a much larger cohort in the peer-reviewed journal Cell. And that's everything I have to present today. Do you guys have any questions?